Our traditional method of developing drugs for breast cancer is you find the drug and it gets tested and the dose gets determined and the toxicity gets established in the phase 1 setting. Then you give it to a few more patients to see whether there's any evidence that it works. If there's evidence that it works, then it gets moved to a larger randomized setting, and those are the traditional registrational phase 3s, where if it does what it's supposed to do and in a way that the FDA approves, then the drug becomes gets FDA indication for that use. That's the point we typically take it into the adjuvant setting. The problem with that traditional paradigm is it's really slow. It takes thousands of patients to answer the questions, and you can only answer essentially one question at a time. So in the modern era of breast cancers being subsetted into multiple distinct biologic subtypes, and we recognize that breast cancer is a lot of different diseases. So that kind of crude way of approaching getting drugs out to our patients is probably not going to work anymore, which is why you're hearing a lot of discussions of alternative designs, designs that let us be more nimble, either by upfront enriching for kind of biomarkers that predict the drug working, or switching drugs more quickly based on a faster readout than you get in a big study, or ignoring whether the cancer came from breast, lung, colon, liver, thyroid, and just saying if it's got this genetic problem, we're going to test this genetically directed drug, and that opens up the arena. I suspect that the future of our ability to develop drugs is going to be much more agnostic about the kind of tumor it is. I also think it's going to require much more tight partnerships between clinicians and scientists, so that they're sort of co-developing the trials. That's really hard to do. It's a very complicated thing, but I think it's the only way we're going to be able to move this in the future.